So the next speaker is Luiz Andrioli, and he's going to talk about how Tailless repression sets up pair rule stripes and gap domains.
Okay, good afternoon. I would like to thank the organizers to be here and have a chance to talk about the work we have been doing in my lab in Brazil. We are presently at EASH, which is a new campus of the Universidade de SãPaulo. I have to say it has been challenging, a lot of work besides science, but hopefully in the end we'll be okay. So in the lab, our main goal is to study regulatory transcription mechanisms during development, and our main work has been with segmentation in flies. This sketch, just to remind you, maternal factors trigger the segmentation cascade, as you can see here, early during the syncytial blastoderm, and the zygotic genes, the segmentation genes are brought, the segmentation genes are progressively refine the expression patterns, leading to the specification of the segments. Our focus in the lab is the interaction among anterior gap genes and the formation of stripes 1 and 2 of the 7-stripe pattern of pair rule genes, such as even skip shown here. Today, I will present the hypothesis we have been investigating in the lab, which is the existence of a repression network regulating anterior pair rule stripes. This hypothesis came from studies with the if stripe 2 regulation. You are probably familiar with that. Becoid and hunchback are broadly expressed in the anterior half of the embryo and able to activate the regulatory element responsible for if2, and the repressors giant and cruple respectively set the anterior and posterior border of this stripe. This is an elegant model. It still explains if stripe regulation, but the way it was first proposed, it's a simplification. For instance, in embryos lacking giants, we can see an expansion of if2 reporter stripe as expected, but according to the model, in the presence of the activators and in the absence of the anterior repressor, we might expect a bigger expansion. So this indicates that one possibility, the other repressors might be working with giants for this repression. To make a long story short, we later showed that the gap domain of slop pair 1 is able to repress if2 in an anterior subdomain, and with giants, it contributes for setting the anterior border of if2. However, still giants and slop are not sufficient to account for if2 repression, and we propose that other repressors might be working as well. At this point, we reasoned that this repression network might be critical for other anterior pair rule stripes. Actually, the anterior-most pair rule stripes of hairy, Eve, runt, and foots. These stripes are formed in a transition zone of a region making stripes and a region that is not making stripes. Indeed, becoid or hunchback were shown to be genetically shown to be activators of these stripes, and nothing was known about their repressors, although regulatory elements were isolated for them. And as you can see, there are many gap domains expressed ahead the striped pattern, and candidates to be repressors for these stripes. We previously showed that slop pair 1, besides repressing if2, also is a common repressor for the anterior-most stripes of hairy, Eve, runt, and foots, as well as Herkbein. And today I will show data presenting a possibility that it's also a repressor for hairy 1 and Eve 1. We use reporter stripes to investigate hairy 1 and Eve 1 expression pattern in different genetic backgrounds. And doing this, we can see subtle but consistent expansions of Eve 1, hairy 1 here, not finding the pointer, and Eve 1 here, as well as a bigger expansion of these stripes in the double mutant, giant and tailless double mutants. And these stripes are also anteriorly shifted. These results show that Tailless might be a repressor for hairy 1 and Eve 1. Moreover, the increased effects in the double mutant show that Sloppy and Tailless might be acting in an additive manner to correctly set the anterior boundaries of these stripes. We also looked at the endogenous stripe pattern of hairy and Eve, and we confirmed our results. And in these experiments, we also detect the expression pattern of other gap genes, such as Herkbein and empty spiracles. And the idea was to make easier the interpretation of the striped pattern results and also to check what's going on with the other gap genes. And we can see here that empty spiracles has a larger domain in tailless null embryos, and this is consistent with previous report in the literature that showed tailless repression on empty spiracles. And you can see also in sloppy null embryos a smaller domain. But what I want to point here is that we didn't find any reasonable explanation that could imply empty spiracles in the regulation of or causing this patterning deviations of the stripes. For instance, if one in tailless null embryo and in sloppy null embryo show a similar problem in their positioning, although the empty spiracles gap domain is very different. So with the endogenous pattern, we also it was also possible to measure the relative position of stripes in the embryos, and for this we developed a computer program which we called morphological analysis tool, and this program coupled to a statistic test mostly confirmed the results we had by visual inspections, which was good, as the effects are really subtle. We also test the tailless repression, and for this we use Gene Expression system. And as you can see, the arrow is pointing. We created a tailless ectopic domain at the ventral of the embryo, crossing the stripe pattern that's being formed. And when we look to the reporter stripes, we detect repression at the ventral parts of hair 1 and Eve 1, and these effects were increased in embryos carrying two ectopic constructs of tailless, suggesting that concentration levels are important for this repression. Very surprisingly, we detect repression for hairy 5 and Eve 5. I have to say that at the time we did these experiments, these were available lines, and hairy and Eve stripes 1 and 5 are contiguous regulatory regions in the loci of these genes, and actually we thought it would be good to use them as internal controls. And we are very surprised when we detect this strong repression for both of them. To be fair, we detect other repression effects for other Peru stripes as well as posterior gap genes. I'm not going to show these results today, and we are presently investigating what's going on. But for Harry and Eve1, we, as I mentioned before, we detect the expression pattern of other anterior gap genes. And besides antispiricles, we also detect patterning deviations for buttonhead. For instance, in Tailless mutant, we can see a larger expression domain of BTD, and consistent with a Tailless repression on this gap gene, and we confirm that with the mis-expression system when we detect a strong repression of BTD. But again, like the situation I mentioned for antispiricles, we didn't find any reasonable explanation implying BTD in the causing the effects we detect for the Peru stripes. In fact, we are getting strong support for direct regulation of Tailless from the genome-wide assays. And here we have the results of two works, one from Noyes and collaborators, showing that there are strong or significant binding sites for Tailless on Harry1 as well as Eve1. And another independent study of MacArthur and collaborators, we also have the same situation. So, today I present what we think is a repression network regulated anterior Peru stripes. Tailless might be part of this repression network. At least we have evidence showing it contributes for the correct positioning of Harry1 and Eve1 anterior borders. And also, as I mentioned before, we detect problems for other stripes and gap genes with the mis-expression system, and we are getting all data from literature, our own results, to see what is going on with these results. And also we are in the process to get triple mutants, which I think is the more decisive way to test our hypothesis. And our goal then is to start in vitro and in vivo experiments to assay, to investigate Tailless repression at DNA level. And I would like to end the knowledge people that are presently in the lab and the collaborators, a great team of bioinformatic people we have at EASH, and Stephen Small, where part of this work has been done. So thank you. If I have time, I'll take questions.
So you said that you're going to do in vitro assays, but have you looked at the sequence? Do you see Tailless binding sites in your enhancers?
From these genome-wide studies, we don't have the results are not refined at the point to show binding sites. We have, there are position wave metrics that we can use, but I was not very confident. And now with these guys, I hope we can, they can help me to better decide this before I start bench work.